The fading of human memory is traditionally viewed as a permanent sentence, an inevitable consequence of the passage of time. Science has long held that the deterioration of the hippocampus is the result of unavoidable wear and tear. Under this consensus, once the brain begins to age, the damage is final. This physical toll is visible at the cellular level. Neural connections weaken, learning capacity slows, and the complex cognitive networks that define our intelligence slowly power off. We have largely surrendered to this decay, treating the loss of our cognitive faculties as a non-negotiable tax on a long life. However, researchers at the University of California, San Francisco began to question the wear and tear model of the aging brain. The team hypothesized that an active biological mechanism suppresses brain function as we age, suggesting that the brain isn't just wearing out, it is being systematically shut down. This shift in perspective allowed the researchers to search for a specific, tangible source of this biological interference. The investigation began by scanning the genetic and protein makeup of the hippocampus in mice, comparing young subjects against older ones. Amidst a vast sea of biological data, one specific protein stood out. It consistently flooded the older, failing brains while remaining nearly absent in the young. This protein is called FTL1. High concentrations of FTL1 correlate precisely with the destruction of neural connections and significant failures on memory tests. Identifying FTL1 transformed the study of brain aging into a search for a specific chemical target that could potentially be manipulated. To test if FTL1 was the actual driver of decline, researchers artificially increased its levels in young, healthy mice. The results were immediate. Youthful neural networks withered, complex branching structures broke down, and young mice displayed cognitive impairments of older animals. To reverse the process, researchers targeted older mice, reducing their high FTL1 levels. The brain physically rebuilt lost connections and restored memory. These tests demonstrate that cognitive decline is a dynamic biological sequence that can be moved in both directions. How does a single protein exert enough influence to dismantle and then rebuild a neural network? FTL1 acts as a metabolic brake. It slows the production of energy within individual brain cells. Maintaining intricate neural connections is biologically expensive. Without sufficient energy, the brain cannot sustain its complex architecture and begins to power down. Lowering FTL1 levels releases this brake, allowing the natural cellular engine to resume the energy production required to power the cognitive network. While this research is currently confined to mice, it establishes a clear path for developing future human therapies. This represents a massive shift in how we understand the mind. Cognitive aging appears to be a manageable biological process rather than an irreversible certainty. We are entering an era where a fading memory might be a condition that can be treated and restored. Growing old no longer has to mean losing the memories and the mind that define who we are.